Welcome to Chapter 16 about DNA and DNA replication. First, we're going to talk a little bit about an experiment that Frederick Griffith did in 1928, showing that there was some form of genetic information that could be passed from one cell to another. Here's what Griffith did. First, he took some living cells that were virulent. This is the S strain here. And when he injected those cells into a mouse, the mouse died. Next, he took some living R cells. These are the non-virulent cells. And when he injected those into the mouse was just fine. Then what Frederick did is he took the S cells, the virulent ones, and heat killed them. He heated them up until they were dead and then injected them into a mouse, and the mouse was fine because the cells were dead. Here's the interesting part. When Frederick mixed the living R cells, which remember didn't hurt the mouse, and the heat-killed S cells, which also didn't hurt the mouse, now the mouse died. And so what Griffith was showing here is bacterial transformation, that the DNA from the heat-killed S cells actually got transmitted to the living R cells, and the R cells were able to actually express those genes for virulence and kill the mouse. So we're going to be doing bacterial transformation in lab, and this is actually how we learned about the genetic information that can change a bacterium from one form into another. Another important experiment to know about is an experiment that Hershey and Chase did in 1952. Now, at this point, it was known that genetic material could be transferred from one cell to another, and that genetic material could be transferred from a virus, specifically a bacteriophage, which is a virus that infects bacteria, into the bacteria. But at this point, nobody knew what kind of molecule that genetic information consisted of. Was it the proteins? Was it the DNA? Hershey and Chase set out to find out. So they took one batch of phage, these viruses that infect bacteria, and grew them up with radioactive sulfur. Now, as you know, sulfur is found in proteins, and so these phage had radioactive proteins. When the phage infected the bacteria were tested and found to be not radioactive. There was no radioactivity in the bacteria themselves. Next, Hershey and Chase grew up a set of bacteriophage with radioactive phosphorus. And phosphorus, as you know, is found in the phosphate group of the DNA. So these phage had radioactive DNA. And when Hershey and Chase mixed these phage with the radioactive DNA with the bacteria, and then rinsed off the extra parts of the phage that did not get injected into the bacteria, they found that the bacteria themselves were in fact radioactive, showing that it was the DNA and not the protein that the bacteriophage were injecting into the bacteria. And this demonstrated pretty conclusively that DNA is actually the molecule of genetic information. And here's DNA. We've seen this before when we studied macromolecules in Bio 101. You can see the sugar phosphate backbone, and each of the sugar groups is attached to one of the four nitrogenous bases, a T, an A, a C, or a G. The combination of phosphate, sugar, and nitrogenous base makes up the nucleotide. So DNA is just a long chain of nucleotides. And nobody really understood how this long chain of nucleotides could be carrying the information of genetics. Now, one of the clues that we had was determined by a scientist named Chargaff. And Chargaff figured out this rule. Chargaff's rules showed that in any species, the number of A nucleotides and the number of T nucleotides is the same. Also, the number of G and C is the same. So for every A, there's one T. For every G, there's one C. And this was an important clue in figuring out the structure of the DNA molecule. So, Watson, Crick, Wilkins, and Wilson, I think I got that right, actually determined the structure of the DNA and that it's a double helix, that it's two curls of DNA with their nucleotides cross-linked at the nitrogenous base. So here on the left side, you can see the overall DNA double helix structure. And then if we zoom in on the right side, you can see here how if a T is sticking off into the center of this double helix, it will hydrogen bond with an A on the other side. And similarly, a G will hydrogen bond with a C. And so each of these nucleotides will match up with its complementary nucleotide on the other side of the double helix strand. So here's a quick activity for you to do. Make sure you've got how this base pairing thing works. Imagine that you have a strand of DNA with this sequence: AA, CG, TGG. So when DNA is replicated, each strand can act as a template or a base for building a new strand in the process of replication. And so here's how it works. On the left side there, A, you have the parent molecule, which is shown in dark blue, the original DNA strand. Now this original DNA strand comes apart so that the double helix is divided into two separate strands. And then each of those two strands can form a new double helix, a new double DNA strand, by fitting in the nucleotides that pair with the template strand, just the way that you put puzzle pieces together. And now you have daughter DNA molecules, each of which consist of half old DNA and half new DNA. So this is known as the semi-conservative model, meaning that each of the new molecules is half old and half. So how does this replication work? Well, let's start by looking at how it works in the very simple chromosome of a bacterium. Now, a bacterium has a round chromosome. It's a single loop of double-stranded DNA, and it has a single origin of replication, one place where the replication process starts. So, in a bacterial chromosome, a replication bubble forms when the two strands at the origin of replication pull apart and new daughter strands are formed using each of those two strands as a template. Now the new daughter strands are built in both directions so that the replication bubble gets bigger and bigger and bigger until eventually you have two new daughter DNA molecules, each of which is a loop and each of which consists of half old DNA from the original chromosome and half new DNA that was fitted into place during the replication process. Now, the way this happens in our chromosomes is a little bit more complicated because our chromosomes are linear, not a loop, and our chromosomes have multiple origins of replication. That is, that the replication process starts at many different points along the linear chromosome. So each of those origins of replication forms a replication bubble, and the replication proceeds in both directions out away from the origin of replication until the growing strand meets up with the section of growing strand that was formed by the next origin of replication. And so eventually you have, similar to the previous slide, two new chromosomes, each of which is made up of half old DNA and half new DNA. So that's the overall process of DNA replication in a nutshell. Now let's zoom in and see at a molecular level what's actually happening as those replication bubbles expand and the new daughter strand is formed. So first of all, it's important to know some useful terminology. The replication fork is that Y-shaped region where the two old strands are pulling apart and the two new strands are getting longer. Helicase is an important enzyme because it unwinds the double helix so that the two strands can be pulled apart at the replication fork. Topoisomerase is an important enzyme that corrects the problem of overwinding. So imagine that you had a ribbon and it was a curly ribbon and you were straightening out one end. As you straightened out one end, the other end would get wound more and more and more tightly. If we have time, we'll do this demonstration in class so you guys can actually see this. What topoisomerase does is it breaks the DNA molecule upstream. So here we have some important enzymes doing their job. Primase, which we'll talk about in a minute, actually building the beginning of the new strand. Helicase is there, pulling the two strands of the double helix apart, and topoisomerase up ahead, keeping us from getting overwound. Now, there are two DNA polymerases, which are the actual enzymes that catalyze the elongation of the new DNA strand. So what DNA polymerases do is they add new nucleotides to the 3' end of the growing strand. The book talks about the difference between DNA polymerase 1 and DNA polymerase 2, but we're not going to focus on that. You just need to know that DNA this is just zooming in to see how DNA polymerase adds new nucleotides to the growing end, the 3' end of the new growing strand. So the nucleotides are actually in the form of a nucleoside triphosphate. They have those extra phosphates added for energy, and then the DNA polymerase pops it into place and the extra phosphates fall off. So mainly you just need to remember here that DNA polymerase can take a nucleotide that's floating around in the cell, pop it into place, matching the template strand. So you see here we had an A on the template strand, so we're bringing in a T, which is the complementary nucleotide, on the new growing strand, and pops it on to the 3' end of that new growing strand. So what this looks like at the replication fork is there you can see on the upper side, the dark blue is the template strand, the old DNA. We have a little RNA primer that's shown in red that was built by primase, and then once that's in place, the DNA polymerase, which is shown in kind of a pinkish color, can come along and pop new DNA nucleotides into place one after another as helicase unwinds the strand so that the new strand grows and grows and grows and grows. So that's what's happening here in our replication bubble. And you can see what we were focusing on just there is here called the leading strand. That's the top left part of the new growing strand. And you can see the leading strand grows all in one piece. It starts with a primer and then just grows and grows and grows and grows. But opposite the leading strand is the lagging strand. Now remember, DNA polymerase can only add to the 3' end of the growing strand. So on the leading strand, that's the end that's growing. No problem. It just keeps adding 3' to the 3' end until it's done. But on the lagging strand, the new DNA needs to grow at its 5' end, and it can't do that. A DNA polymerase can't add any nucleotides to the 5' end. So what happens instead is on the lagging strand, the new daughter strand grows in the opposite direction that replication is actually taking place in a series of smaller fragments that eventually get joined together. So let's zoom in and look at what's happening on the lagging strand. So here we have a lagging strand. Up at the top you see the template strand. We build a primer, and then DNA polymerase is able to add to the 3' end of that primer, going from left to right. And the little piece of DNA that gets built in that process is called an Okazaki fragment. Then, farther to the left, the direction that replication is moving, a new primer is built and a new Okazaki fragment is built until it reaches the previous primer. At that point, the RNA of the primer is replaced by DNA, and the 2 DNA strands are eventually joined together by ligase to form one single new daughter strand. So it's important to keep in mind all of these different enzymes that are helping. Helicase, the single-strand binding protein just keeps the single-strand DNA stable. Topoisomerase, which keeps the strand from getting overwound. Primase is actually building a new RNA primer, one on the leading strand and one for each Okazaki fragment of the lagging strand. Then we have the DNA polymerases, which bring in DNA nucleotides to grow the new strand. One of them grows the new strand when it's just free. The other one can actually replace the RNA of the primer, but don't worry too much about that. The important thing here is that DNA polymerase adds new DNA nucleotides to the growing strand, and then ligase joins one Okazaki fragment to the next Okazaki fragment. So here you can see all those enzymes doing their work on the lagging strand. The primase is building a primer, and then DNA polymerase is bringing in those new nucleotides, first in the gaps and then later to replace the previous primer, and finally DNA ligase is attaching those fragments together. And that's how replication occurs on the lagging strand, one fragment at a time. So in this diagram, replication is happening overall from right to left, right, heading toward the replication fork there. But each of those Okazaki fragments is being built from left to right because DNA polymerase can only add to the 3' end of the growing strand. Now, as it's going along, DNA polymerase is proofreading the new DNA and replacing any incorrect nucleotides. So if you've got a C on the template strand and a G, so here's a demonstration of how that would work. In this case, two nucleotides have gotten bonded together in a way that they're not supposed to be. So nuclease comes in, it cuts out that section that's damaged, and then using the other side of the double helix as a template, DNA polymerase can pop in the nucleotides that belong there, and then ligase once again glues the new sections together so that now we have a fixed section of double-stranded DNA. Now, what happens when we get to the end of a linear chromosome? Remember, unlike bacteria, your chromosomes are linear. They're a straight line, not a loop. On the leading strand, no problem. DNA polymerase just keeps adding new DNA nucleotides until it gets all the way to the end and falls off. No big deal. But on the lagging strand, we have that final primer. And there's no way for those RNA nucleotides in that last primer, that last little 5 to 10 base pair section, to get replaced because there's no way for DNA polymerase to add in those new DNA nucleotides because it doesn't have a 3' end of the growing strand to grab onto. So what happens is every time DNA is replicated, a little bit of the lagging strand gets chopped off. I'm going to say that again. Every time a linear chromosome gets replicated, a little bit of the lagging strand gets chopped off. It gets shorter and shorter in every replication. So this is just a repeat because DNA polymerase cannot complete the 5' ends, so every time a replication occurs, the DNA molecule gets a little bit shorter. Luckily, at the ends of our chromosomes, we have telomeres. Telomeres are actually part of the DNA, part of the chromosome, but it's a part of the chromosome that's just nonsense. It's basically garbage DNA at the end of our chromosomes. What that means is, even though our chromosomes are in fact getting shorter and shorter with every replication, we're not actually losing genes. We're not actually losing important DNA. We're losing this junk DNA that's found at the end of each chromosome. So telomeres, these sections of junk DNA that you can see are highlighted in this image, are there to prevent the actual genes from getting shortened. Now, eventually you run out of telomeres, right? You've used up all of your junk DNA, and it's been now, if the chromosomes of your germ cells, these are the cells that produce eggs and sperm, became shorter in every cycle just like the rest of your cells, then your eggs and sperm that you produce would have the same short telomeres as the rest of your cells. It would be like if you were 25 when you had a baby, the baby's cells would already be 25 years old. Obviously, that's not a good situation. And so we have an enzyme called telomerase. Telomerase builds new telomeres. So in the germ cells, the cells that are responsible for producing eggs and sperm, telomerase actually lengthens those chromosomes by adding extra junk DNA at the end. Now, it's good to have telomerase in your germ cells because it basically provides a reset button so that new babies are born with long telomeres instead of short ones. And that's great. They're born basically at age 0. But you don't want to have telomerase in the rest of your cells because the shortening of your telomeres actually acts as a stop to cancer. Remember, cancer is just one cell that replicated over and over and over again more times than it should. In most cases, this can't happen because if a single cell replicates over and over and over again, it will eventually run out of telomeres, right? Run out of that junk DNA and no longer have functional chromosomes and die. And that's what you want. You don't want any one cell to reproduce over and over and over again because that's cancer. And there's been some evidence to suggest that telomerase may actually be present in cancer cells and helping them to grow new